Welcome back to one more video. In this video we are going to discuss about beta-oxidation of the fatty acids. It is one of the important metabolic pathways of lipid. It's called the beta-oxidation because the beta carbon of the fatty acids undergoes oxidation at one point. But before we start this process, what is a fatty acid? A fatty acid is nothing but a long chain of the carboxylic acid having at least 4 to 36 carbon in its long chain. There are two types of fatty acids, the saturated fatty acids and the unsaturated fatty acids. The saturated fatty acids do not have the double bonds and unsaturated have the double bonds. Saturated best example is the palmitic acid, which is a 16 carbon carboxylic acid without any double bond. And unsaturated best example is linoleic acid, which is an 18 carbon carboxylic acid having at least one double bond at 9th position. These unsaturated fatty acids exist liquid at room temperature, so they are termed as oils. And the saturated fatty acids exist solid at room temperature, so they are termed as fats such as butter and ghee. Once when we look at our dietary lipids, the lipids we take in our diet, such as this delicious pizza having the cheese and the mozzarella. The lipids that we take in our diet, 98% of them are in the form of triglycerides or triglycerols. Triglyceride means having one glycerol head and 3 fatty acid chains. And these fatty acid chains may be of unsaturated or saturated type, and these are simple lipids. The triglycerides having the saturated fatty acids are stored in the form of body fat in adipose tissue. These triglycerides, which are stored in the form of body fat, have to be broken down into their free forms for the beta oxidation. For this, the enzyme lipase acts on them and catabolizes them into the glycerol head group and the free fatty acid tail group, which are hydrophobic. The above glycerol can be phosphorylated into glyceraldehyde triphosphate, which can then enter into the glycolysis in the cytoplasm to produce pyruvate, and from the pyruvate to produce acetyl-CoA. Acetyl-CoA can also be produced by the free fatty acids by the beta oxidation in the target cell and then can enter into Krebs cycle. But for the beta oxidation, first it should be transported into a target cell through the bloodstream. In the bloodstream, we have a special transporter called the serum albumin transporter, which can transmit this free fatty acids into a target cell for the beta oxidation. But remember, this target cell can be any cell except a mature RBC and also not a neuron, because a neuron and RBC do not have the mitochondria. So the beta oxidation will not take place here. So that's how the overall transportation of the fatty acids into target cell. Once recap, the glycerol from the body fat can enter into the glycolysis to produce pyruvate and acetyl-CoA. Similarly, the free fatty acids, the hydrophobic parts, can be transported into a target cell through the bloodstream, and the bloodstream through the serum albumin, they can be transported into a target cell except RBC and neuron. The target cell can be a liver cell which performs the beta oxidation and can convert this into acetyl-CoA. In acetyl-CoA can be used by Krebs cycle to produce NADH, FADH2 and ATP. So that's how the fatty acids are catabolized from the body fat and enter into target cells for beta oxidation. But what actually happens in a target cell? Now the fatty acids have been brought near the target cell through the blood serum albumin transportation, and this target cell has the mitochondria for the beta oxidation. Let us zoom this part of the cell here. In order for the fatty acid to cross the plasma membrane of the target cell, this fatty acid, the palmitic acid, to enter into the cytoplasm of the target cell, it has to be activated into an acyl form. So this palmitic acid is converted into palmitoyl-CoA, an acylated form by adding CoA. In order for the palmitoyl-CoA to perform the beta oxidation, it should enter into the mitochondrial matrix by passing outer and inner mitochondrial membranes. But in order to transfer this palmitoyl-CoA into the matrix of the mitochondria, we have a carnitine shuttle because the inner mitochondrial membrane is impermeable because of the cardiolipin. Now the palmitoyl-CoA in the cytoplasm of the target cell first need to be transported in the periplasmic space. This is done by the carnitine palmitoyl translocase 1 located in the outer mitochondrial membrane, which transports this by adding carnitine and removing CoA and converts it into palmitoyl carnitine. But in order for this palmitoyl carnitine should be transported from the periplasmic space into the matrix, it has to cross the inner mitochondrial membrane, which is completely impermeable. For this, we have a special type of transporter called Cn-acyl-CoA translocase 2, which transports palmitoyl carnitine from the periplasmic space into the mitochondrial matrix. But this carnitine have to be pumped back into the periplasmic space. For this, the located palmitoyl carnitine translocase 2 transports the carnitine back and forming the palmitoyl CoA. The carnitine can be reused in this process. This entire thing is called as a carnitine shuttle. This entire mechanism is called carnitine transport system, and the substance used here is L-carnitine. For your body to produce this L-carnitine, you need sufficient amount of vitamin C, so don't neglect your vitamin C. So, the palmitoyl-CoA from the cytoplasm is transported into the periplasmic space and from here to matrix, and the carnitine from the matrix is retransported in the periplasmic space through the carnitine transport system. So the carnitine does makes an important role in the transportation of the fatty acids, and this repeats again and again and again. So after the beta oxidation, the fatty acids can yield acetyl-CoA, which can be utilized by the Krebs cycle to produce ATP. So that's all about the part 1 of this video. In next video, we will see the actual mechanism of the beta oxidation. Thank you for watching. Please do subscribe.